Our Earth, a delicate and interconnected web of ecosystems, relies on healthy soil to sustain life. However, the silent menace of soil pollution has been steadily infiltrating this vital resource, threatening the delicate balance of our environment. In today's episode, we explore how human-induced pollution reshapes the intricate relationships within soil ecosystems and examine the responses of soil when subjected to such conditions. Are you ready to dive into the hidden world beneath our feet?
Currently, chemical pollutants and plastics are two primary causes of pollution in the soil ecosystem. Firstly, chemical pollutants such as petroleum hydrocarbons, including crude oil, introduce a complex mix of hydrocarbons into the soil. When available underwater, petroleum can lead to seeps, fostering microbial blooms exploiting this resource. Also, per and polyfluoroalkyl substances known as forever chemicals present a contemporary challenge due to their resistance to degradation. Additionally, halogenated organic molecules like PCE, TCE, and PCBs further complicate soil ecosystems, demanding innovative bioremediation strategies. Lastly, as we are all familiar, plastics designed for durability have become a global environmental issue. Their prolonged decomposition rate and resistance to biodegradation pose a threat to soil ecosystems. The long hydrocarbon chains in their structure make it challenging for microbial enzymes to break them down.
But wait, let's first go back to the root of all of this. Stay tuned as we unearth the secrets concealed beneath the soil's surface.
Imagine a world beneath the Earth's surface, a hidden community bustling with activity. This is soil, a complex matrix comprising microorganisms, organic matter, minerals, and gases, all working together in harmony. It is within this rich habitat that an extraordinary diversity of microorganisms thrive, shaping the very fabric of our planet. In the depths of soil, an enchanting cast of characters reside. Bacteria, actinomycetes, fungi, algae, protozoa, archaea, each with its unique role to play. These microscopic heroes are joined by viruses and other remarkable eukaryotes like fungi, adding to the tapestry of life beneath our feet. But that's not all. Soil is not just a passive recipient of life's grand tapestry, but an active participant in the health and vitality of ecosystems. It is the foundation upon which our forests, grasslands, and agricultural systems flourish. Through the lens of soil microbiology, we uncover the vital connections between soil and plant health, the fascinating mechanisms of disease suppression, and the potential for sustainable agriculture.
We will now uncover the mysteries, unveil the wonders, and deepen our understanding of the captivating world of soil microbiology, a field that holds the key to understanding how microorganisms in our soil can combat the looming threat of soil pollution. But first, what is soil microbiology in general?
Soil microbiology, simply put, is the study of this tiny, mighty organism that make the soil not just dirt, but a living, breathing entity essential for the balance of our ecosystem. It's a branch of microbiology dedicated to unraveling the mysteries of microorganisms in the soil and their interaction with the environment. Soil microbiology is very crucial for nutrient cycling, organic material decomposition, and maintaining soil fertility. It is basically the engine that drives the flourishing life above ground.
But what happens when said tiny organisms encounter the challenge of pollutants and organic compounds?
So, one of the important aspects of soil microbiology is biodegradation. It is a natural microbial-driven process that catalyzes soil cleansing and rejuvenation.
Biodegradation, a fundamental process in soil microbiology, involves the natural breakdown of organic chemicals through the action of microorganisms. These soil microorganisms, which include bacteria, fungi, and other microbial communities, work together in a consortium to carry out biodegradation.
In the world of soil microbes, there's a team of tiny creatures working together. These microbes release special substance called enzymes, which are like their superpowers. These enzymes are expert at breaking down complex substrates in the soil. Imagine the soil as their playground, and these microbes play a game of breaking down things like oil or hydrocarbons, enzymes, pesticides, and metals that can harm environment. They have this amazing ability to turn these harmful things into safe and friendly stuff.
Biodegradation has three general steps. These are biofragmentation, bioassimilation, and mineralization. In the first part of the breakdown process called biofragmentation, large and complex organic compounds get broken down. Microscopic organisms release special enzymes called exoenzymes into their surroundings. Think of these enzymes as tiny molecular scissors. Their job is to cut those complex organic molecules into smaller and simpler pieces. This step is really important because it makes the organic material smaller and easier for other tiny organisms to work with in the next steps. After the big molecules are broken down in the first step, we move on to the next phase called bioassimilation. Here, tiny organisms absorb the smaller pieces produced during the breakdown. These bits could be things like peptides, amino acids, sugars, and other simple organic stuff. Now, these tiny organisms use these absorbed bits like a power snack. It's like their favorite energy bar. They eat up these pieces, and through some fancy chemical processes inside them, they turn these snacks into energy and nutrients. It's like a tiny feast that helps them grow, reproduce, and do all the things they need to do to stay alive and well. In the final part of biodegradation, we reach mineralization. This is like the last stop where the transformation of organic stuff into simpler inorganic forms happen. During mineralization, any leftover organic compounds are broken down even more into their most basic parts. For instance, think of carbon-containing compounds. They get oxidized and produce carbon dioxide, kind of like when you breathe out. At the same time, other inorganic nutrients like nitrogen and phosphorus are released. These nutrients are like the building blocks that plants and other living things need to grow. So, mineralization is like the grand finale in the biodegradation show. It wraps everything up by turning the remaining bits of organic material into simple and essential forms like carbon dioxide and nutrients. These get put back into the environment, ready to be used again by other living things. It's nature's way of recycling and making sure nothing goes to waste.
Now that we've explored the fascinating world of biodegradation, let's embark on a journey to discover how this natural process seamlessly intertwines with another essential aspect of soil health, nutrient cycling. Biodegradation, as we learned earlier, is nature's recycling system. But what happens to the byproducts of this process? Well, that's where nutrient cycling comes into play. Exploring the carbon cycle is vital for comprehending global climate change. Identifying sources, sinks, and reservoirs of carbon dioxide is essential. Heterotrophic microorganisms and fossil fuel combustion release carbon into the atmosphere, while plants, phytoplankton, and autotrophic microorganisms act as sinks, storing carbon in various forms like methane and carbon dioxide.
Carbon fixation, the conversion of carbon dioxide into organic matter, occurs through photoautotrophic and chemoautotrophic microorganisms. About half of the Earth's carbon is fixed by microorganisms, including diatoms and marine cyanobacteria. Microorganisms play key roles in anaerobic photosynthesis, chemical lithotrophy, and even deep subsurface carbon fixation. Methane, produced by archaea, originates from organic matter or anaerobic reduction of inorganic substrates. Sources include sediments in various environments like rice paddies, coal mines, and even the digestive systems or termites. Aerobic oxidation and anaerobic oxidation processes play a crucial role in mitigating methane emissions. The nitrogen cycle, more complex than the carbon cycle, involves nitrogen species in various redox states. Nitrogen compounds act as electron acceptors or donors in anaerobic respiration and chemical calcification, influencing their roles in ecosystems.
Nitrogen fixation, performed by bacteria and archaea, reduces N2 to ammonia, crucial for the synthesis of organic substances. Nitrification, the oxidation of ammonium to nitrite and then to nitric acid, occurs aerobically. Recent discoveries like CommaMox challenge previous notions about nitrification processes. Denitrification, carried out by various bacteria, reduces nitrate to nitrogen gas or nitrous oxide, returning nitrogen to the atmosphere. The Anamox reaction, an anaerobic process, provides a shortcut to direct reduction to nitrogen gas, holding potential applications in wastewater management. The biogeochemical cycle of phosphorus is vital for all living cells, serving roles in ATP synthesis, nucleic acids, and lipid production. Phosphorus enters ecosystems through decomposition, weathering, and fertilizer application. Recent studies suggest that phosphonates in organic matter contribute to phosphorus availability. Microorganisms play a crucial role in recycling both inorganic and organic phosphorus, influencing the availability of phosphorus to plants and microorganisms.
The scope of biodegradation is vast, encompassing a wide range of natural processes that occur in various environments. It influences the decomposition of organic matter, ensuring the cycle of life continues in harmony. But when pollution strikes, biodegradation alone may not be enough. This is where bioremediation comes to play.
Bioremediation is the process of using microbes to transform toxic contaminants into non-toxic degradation products. Compounds such as perchloroethylene, trichloroethylene, and polychlorinated biphenyls are the common contaminants targeted for removal.
Bioremediation is like nature's cleanup crew for dirty places. Imagine a polluted area, like soil, with harmful stuff in it. Bioremediation is a way we humans help nature to do its job for breaking down and getting rid of those pollutants. We introduce certain tiny organisms or make the conditions better for the ones already there to speed up the processes of cleaning up the mess. It's like giving Mother Nature a hand to fix the environment when it's been damaged by pollution.
In the field of environmental science, the process of reductive dehalogenation holds great significance. It is a chemical reaction that occurs when a halogen substituent, such as chlorine, bromine, or fluorine, is removed from a molecule while simultaneously adding electrons. This reaction is commonly observed in the presence of certain microorganisms and enzymes that have the ability to break down halogenated compounds.
At first, pollutants are in forms that can harm our ecosystems. But thanks to both natural and human-made processes, these harmful forms can change into less toxic ones that are easier to break down. One process, known as reductive dehalogenation, is particularly involved in this transformation.
There are two main mechanisms that describe for reductive dehalogenation. First is hydrogenolysis. In this process, the halogen substituent is replaced by a hydrogen atom. This involves breaking the bond between the halogen and the carbon it is attached to, and the hydrogen atom is introduced in its place. Second is dihaloelimination. This mechanism removes two halogen substituents from adjacent carbons, creating an additional bond between these carbons. Essentially, two halogen atoms are eliminated, and a new bond is formed between the neighboring carbon atoms.
When it comes to biodegradation and bioremediation of a compound, several influential factors come into play. Two such factors are chirality and isomerization.
Chirality is like a molecule having two mirror image forms. Microbes can break down one form but not the other, and this choice matters a lot in biodegradation. About 25% of herbicides have this characteristic, so picking the right form is crucial for successful cleanup. Isomerization, a process where a compound structure changes, also affects how fast it breaks down. Different versions of a compound can break down at different rates in nature. For example, a compound in a meta position breaks down slower than its counterpart.
Take the herbicide 2,4-dichlorophenoxyacetic acid. With chlorine in the ortho position or adjacent to the primary carbon on a benzene ring, it breaks down fast. but 2,4,5-trichlorophenoxyacetic acid with a different arrangement lasts for years, making it good for long-term brush control.
An emerging approach to the bioremediation of contaminants in soil is the use of microbial fuel cells. A tiny powerhouse in the field of renewable energy comes from the ground we walk on. The anaerobic oxidation of organic materials by a variety of heterotrophic microorganisms has revealed a potential energy solution: the microbial fuel cell, or MFC.
Chemical mediators are used to move electrons across the membrane and to the anode, which solves the problem of efficiently delivering electrons to the anode. This paves the way for a breakthrough that finds use in situations where conventional batteries are impracticable, including the long-term installation of environmental monitoring equipment.
Almost every inch of soil contains bacteria with the power of produce energy when placed in an MFC. The more electron-producing, soil-munching bacteria, the more power the MFC generates. This potential opens doors to clean, renewable electricity in almost any location on Earth, tapping into the abundant presence of bacteria-filled soil.
It's amazing how straightforward the MFC design is. The foundation is made up of two chambers, one anoxic and the other oxic, divided by a membrane. When protons permeate through the membrane and enter the oxic chamber, the flow of electrons created by the oxidation of organic materials generates electricity. The microbial fuel cell's ability to produce electrons makes them essential to this energy-producing process. The goal of ongoing developments in MFC technology is to increase power output, expanding its uses to include landfill leachate treatment and drinking water purification.
So there is a potential in using soil in MFCs to produce power. Research explores the complex interplay among exoelectrogenic bacterial species, soil properties, and electrical output. The Geobacter and Schwannella species, which are common in soils over the world, are essential to this change. The combination of environmental microbiology with MFC technology offers a glimpse of a future in which the very ground we walk on generates clean, renewable energy as we explore the untapped potential of our soil. A powerful relationship between innovation and bacteria develops in the microscopic world below, paving the path for a future that is less harmful to the environment.
Before this episode ends, we need to be aware that more challenges in soil microbiology also stem from soil pollution.
Firstly, the impact of climate change on microbial diversity. Microorganisms, the unsung heroes of balanced element cycling, face unprecedented challenges due to global climate change. The alterations in microbial activities have far-reaching consequences on the rates and magnitudes of greenhouse gas accumulation, contributing to the ongoing global climate crisis. Climate-induced disruptions force adjustments, migrations, or even extinction in the relationship of species, and extreme weather events and habitat modifications challenge microbial populations, affecting their distribution and overall diversity. And secondly, soil microorganisms and greenhouse gas emissions. Soil microbes are crucial for carbon cycling, a vital process for Earth's biogeochemical cycles. However, the accelerated release of greenhouse gases, such as carbon dioxide and nitrous oxide, surpasses natural recycling rates. Microorganisms break down organic matter and release these gases into the soil during decomposition. The imbalance between gas release and recycling aligns with the rise in global temperature, culminating in the escalating concern of global warming.
In the hidden world beneath our feet, soil microbiology orchestrates a silent symphony essential to life on Earth. These microscopic heroes shape our ecosystems, sustain agriculture, and impact the very air that we breathe. So join us again as we explore the unseen forces that make our planet thrive. Until then, stay curious and keep discovering the wonders beneath the surface.